My name's Chris Tape. I'm a junior group leader at the UCL Cancer Institute in London, and I lead the cell communication lab. And my lab studies how different cell types interact in cancer, so we're particularly interested in how oncogenic mutations in cancer cells interact with non-mutated cells such as stromal fibroblasts and immune cells in the tumour microenvironment. We're also developing a lot of new technologies to analyse these. So we do a lot of single cell analysis because we need cell type specific information in what we're analysing. So we do a lot of mass cytometry and single cell RNA-seq, and we're also developing new single cell proteomic based technologies to do multimodal measurements of a lot of the complex tumour models that we use. So my lab using a lot of organoids. We use organoids from animal model systems such as mice, where we have very controlled genetic environments where we know exactly what genetic events are controlling the phenotypes. But then increasingly, with the more translational ambition, we're using a lot of patient organoids. And in the patient organoids, we're taking tissue directly from patients from a resection, from surgical resection, extracting the organoids from them. Also taking paired stromal cells and autologous immune cells, and using those to study patient-specific responses, because we think in cancer it's not just about a defined mechanism that we can put in a textbook that will be universal for all patients. It's really a patient-specific disease. Every patient is its own kind of unique entity. So by taking patient-specific models, we can do mechanistic work to understand patient-specific mechanisms. Organoids are great model systems. They have pros and cons. I think in the pros column, they would be they're very closely paired to patients, so we get good correlation between patient drug responses and what we might see in an in vitro system in an organoid. They also have a high amount of plasticity, so increasingly in cancer we're interested in non-genetic events that might be controlling a drug response, for example, and some of that can happen through differentiation, and organoids, being stem cell driven systems, have high amounts of differentiation in them, so that allows us to study these non-genetic events. Downsides, they're quite expensive to work with because we have to use matrigel culture and all these kind of complex ECMs, and that also brings a lot of technical complexity. We've got to get information out of the organoids. It's very well growing them in a dish, but then you've got to get information out of them, and they're three-dimensional. They have differentiation effects, they have stem compartments and cell state complexities. So that's where a lot of our single cell technologies that we've been developing really help us to understand that high dimensional complexity of the models. So I think the cancer dependency map is a fantastic resource, and I think it helps researchers all around the world go from observations that they may have made in a more anecdotal setting, so in a couple of model systems that they may have access to in their lab, to then extrapolate and explore those findings across potentially hundreds of cancer cell lines, and how that might relate. And I think that's a very powerful resource, because we can't all do a depth map. We shouldn't all be doing a depth map in our own labs. It's by outsourcing this to kind of a consortium approach, it then allows researchers in smaller labs to leverage that information and to maybe generalize some of the findings they might be making. So, DepMap is already an incredible resource. I think in the future we need to bring in more single cell measurements. I think at the moment a lot of the outputs of DepMap have been very binary, very live dead based through bulk analysis, and I think in the future we'll be needing to do more single cell analysis, whether that's at the protein or phosphorylation level or a transcriptional level. I think that will push the resource on further. And then I really think, and this is my bias as a cell communication person, I think we need to have other cell types in there. We know in biology and in cancer that no cell type sits on its own. Every cell type is surrounded by other cell types, whether it be just in the homeostatic tissue or in the tumour microenvironment. So by having different cell types present during some of the perturbation style screens that we're seeing from DepMap, I think we could get better insight into how these events may function in a wider tissue context, which is ultimately involved in the disease.